Okay, Assalamualaikum warahmatullahi taala wabarakatuh. Good morning everyone. So today we are going to study a new topic, okay? So which is about enzyme. So for those who don't have physical textbook, okay, you can use the online flip over textbook. Okay, I have share with you the link. Okay, you can make use of it. Okay, guys. So today we are going to study about enzyme. Okay. So before we want to study about enzyme, you have to know what does metabolism means. Okay. So metabolism is any chemical reaction that occur in living organism. Okay. So any chemical reaction that happen in our body. For example, the formation of ATP energy, the formation of carbohydrate, the formation of lipid. So any kind of process happen in our body is known as a metabolism. Okay, so basically there are two types of metabolism. The first one is catabolism. The next one is anabolism. Okay, so what is the difference between these two? Okay, the first one, catabolism, is a process of breaking down complex substance into simple substance. So example like this, you have A, so A is a big molecule. So if we want to break them, okay, so it will produce smaller or simple substance which are B and C. Okay, so this process is known as a catabolism. Why? Because it will break down the complex substance which is A into simple substance which are B and C. Okay, so that one is for catabolism. The next one is anabolism. Okay, anabolism is a process of synthesizing complex molecule from a simple molecule. For example, you have simple molecule A and B. Okay, so and then A and B you combine them. Okay, so it become large molecule. So this process is known as a anabolism. Okay, so so for catabolism, it's a process to break down large molecule into small molecule. But for anabolism, okay, it's a process where we have simple molecule, we add them together to produce a large molecule. Okay, so example for anabolism is a process of to produce, okay, or to form glucose during photosynthesis. Okay. Okay, guys. So the next one, we are going to study about enzyme. Okay. So what does enzyme means? An enzyme is an organic catalyst. Okay, catalyst ni maksudnya pemangkin. Pemangkin. Okay, that is mostly made up of proteins and produced by living organism. Okay, so this is how enzyme looks like. Okay, so as you can see, enzyme, it has special structure. Okay, the name of special structure is active site. Okay, so this is enzyme. Okay, so enzyme, it has special structure which is known as a active site. Okay, so active site, what is the function of active site? The function of active site is to enable, okay, the enzyme to bind with substrate. Okay, so this is substrate. Okay, okay, teacher, what does substrates mean? Okay, you see here, this is the definition of substrate. Substance needed for enzyme reaction. Okay, for example, okay, we know that the enzyme, there are many example of enzyme. For example, you have lipase. Okay, for example, your enzyme is lipase. So the function of lipase to break down lipid, right? So our substrate here is lipid. So enzyme is lipase. The substrate will be lipid. Okay, for example, another example for my enzyme is amylase. What should be my substrate? My substrate should be starch, right? Because the function of amylase is to break down starch into maltose. Okay, so when the enzyme, okay, they have active site, right? So this active site will enable the substrate to bind with the enzyme. So when they bind together, this will produce enzyme substrate complex. Okay, so this structure is known as a enzyme substrate complex. Okay guys, so far so good. Okay guys, so let's go to the next one. Now we go, okay, so here they want to share with you how you can name enzyme. Okay, so basically if you want to name the enzyme, you just put ASE at the back. Okay, for example, okay, you have not at, right? Maybe you can change the end of the words. Okay, for example, your enzyme sorry, your substrate is lactose. Okay, so in order for you to know what is the suitable enzyme required for this reaction, you just replace O with A. So ASE, it means lactase. For example, you have maltose. What is the name of the enzyme? Maltase. Okay. So this is how you name the enzyme. Okay. Okay, guys. The next one, you are going to study about the general characteristic of the enzyme. Okay. So let's go one by one. Okay, we go here. The enzyme are only required in a small quantity and are reusable. So you don't have to bother yourself to use a lot of enzyme. Why? Because a liter of enzyme can works the best. Okay, so you require in a small quantity, so do not waste it. And then they can be reused over and over again. Okay, that's why they are reusable. Okay, and the next one, the structure of enzyme will remain unchanged and not destroyed after reaction. For example, you see here, this is how enzyme looks like, right? So when the enzyme react with substrate, okay, the enzyme will remain the same. So this is how the enzyme looks like before and after the reaction. They will stay the same. Why? Because, okay, the structure of enzyme remain unchanged and are not destroyed after reaction. So that's why it can be reused. Okay. The reaction of enzyme are specific. Only substrate that fit the form completely with an enzyme in an active site can combine. Okay, what does reaction of enzyme are specific? Okay, as I said just now, we have so many enzymes, right? We have amylase, we have protease, we have lipase, and so on, right? So for example, you have amylase. So amylase is meant for starch. Lipase is meant for lipid. Protease is meant for protein. So can you see the reaction of enzyme is specific. Okay. Can you use lipase to digest starch? No, right? You in order to digest starch, you require amylase. So we can say that the reaction of enzyme are specific. The specific lah. You know specific, right? Specific itu maksudnya khas untuk tu saja. Okay. Okay, the next one. Most of reaction catalyzed by enzyme are reversible. Okay, reversible. Maksudnya contohlah, you have starch, right? Starch, you use enzyme amylase to to produce maltose. So if you want to use if you want to change maltose into starch back, is it can be do it can sorry it can be done. Why? Because the reaction of enzyme are reversible. Maksudnya dia boleh berpatah balik. Okay, the next one. The enzyme activity can be slowed or stopped by enzyme inhibitor. Okay. An example of inhibitor is a heavy metal such as lead and mercury. So if you have lead and mercury inside your body, okay, this will disturb or this will stop, okay, the enzyme activity, which is not good. Okay. The next one, some enzyme need cofactor to works more efficiently. For example, cofactor are vitamin, okay, magnesium, iron, and so on. So these two will help the enzyme to works better. The next one, enzyme are biological catalysts that speed up the biochemical reaction. So this will make the process of digestion become faster and more efficient. And the last one, the enzyme act rapidly. So the reaction of enzyme will occur faster. Okay. Okay, guys. So that's all for the characteristic of enzyme that you should know. Okay, the next one, we are going to study about intracellular and extracellular enzymes. So bear in mind, there are two types of enzyme. The first one is intracellular. The next one is extracellular. So what does it mean? Okay, so kita tengok satu-satu. Okay, kalau intracellular enzyme is an enzyme that is synthesized. Synthesized tu maksudnya dihasilkan. You can use the word synthesize. You also can use the words produce. Okay, they deliver the same meaning. Okay, enzyme that are synthesized in a cell for its own use are called intracellular enzyme. Okay, for example, awak ada sel, right? Sel awak tu menghasilkan enzim dan enzim tu digunakan dalam sel itu sendiri. Itu masuk intra. Intra tu maksudnya dalam. Apa yang dalam? Maksudnya enzim yang dihasilkan tu dia akan digunakan di dalam sel itu sendiri. Faham? Okay, the next one you have extracellular enzyme. Yang ni awak tukar saja. Yang tadi cikgu cakap, kalau intracellular, sel tu hasilkan enzim dan enzim tu digunakan dalam sel. Betul tak? Tapi kalau yang ni, extracellular enzyme ni, enzim tu dihasilkan dalam sel tapi digunakan di luar sel. Ni awak tengok ni. On the other hand, enzyme that are secreted outside the cell are known as a extracellular enzyme. Maksudnya sel tu hasilkan enzim, lepas tu sel tu dia keluarkan enzim tu sebab apa? Dekat luar enzim tu nak digunakan. Okay? Okay, example dia ni, example dia trypsin enzyme are produced by pancreatic cell and secreted into duodenum to break down polypeptide. Okay, sebab apa? Pancreas yang hasilkan enzim. Tapi pancreas tu tak guna pun. Sebab apa? Pancreas tu akan bagi enzim tu kepada duodenum. So maksudnya enzim trypsin ni tidak digunakan dalam pancreas tapi digunakan di dalam duodenum. Okay, sebab tu di extracellular. Extra means kat luar. Okay. So how extracellular enzyme are produced? Macam mana extracellular enzyme are produced? The production of enzyme involve a few specific component of the cell. Okay guys, so this is the mechanism or process how extracellular enzyme are produced. Okay, kita pergi satu-satu. Okay, before we want to look at this diagram, okay, I want to introduce to you the structure inside the cell. Okay, so yang dalam yang kuning ni, okay, yang kuning awak tengok ni, this is inside the cell, dalam sel. Okay, yang warna biru ni, this is outside the cell. Okay, sekali lagi yang kuning ni dalam sel, yang warna biru ni luar sel. Okay, so this is nucleus. Okay, yang ni rough endoplasmic reticulum. Ni rough endoplasmic reticulum. Okay, rough endoplasmic reticulum ni dia ada bulat-bulat ni awak tengok ni. Biji-biji ni kan. So bulat-bulat ni nama dia adalah ribosom. Okay, bulat-bulat ni nama dia ribosom. Ini rough endoplasmic reticulum. Okay, and then on its surface there are many ribosome. Apa fungsi ribosom? Fungsi ribosom is to produce protein. Okay, the function of ribosome is to produce protein. Okay, boleh faham? Okay, the next one, awak ada Golgi apparatus. Okay, awak ada Golgi apparatus. Apa fungsi Golgi apparatus? Fungsi Golgi apparatus is to modify. Ataupun ubah suai. Is to modify protein into enzyme. Okay. Cikgu ulang balik. Okey, macam ribosom ni, fungsi dia adalah untuk menghasilkan protein. Okey, kalau Golgi apparatus fungsi dia adalah untuk menukar protein tu. Tukar protein kepada apa? Kepada enzim lah. Kita kan nak menghasilkan extracellular enzyme, betul tak? Okey, itu saja. Okey, untuk tu. Yang lain cikgu explain kemudian. Okey, jom kita pergi satu-satu. Nombor 1. Ribosome is the site of protein synthesis. So kita boleh cakap pertama sekali, ribosome ni adalah tempat yang mana kita akan hasilkan protein. So protein synthesized by ribosome enter to the lumen of rough endoplasmic reticulum and transported through it. So maksudnya ribosome ni dia akan hasilkan protein. So protein tu dia akan masuk ke dalam rough endoplasmic reticulum. Sebab apa? Rough endoplasmic reticulum ni dia akan hasilkan transport vesicle. Tengok ni. Okay, rough endoplasmic reticulum, it will produce transport vesicle. Sebab apa kena buat transport vesicle ni? Sebab protein yang dihasilkan oleh ribosom tu need to be transfer to the Golgi apparatus. Sebab apa kena transfer ke Golgi apparatus? Sebab kita nak tukar protein tu kepada enzim. Betul tak? Okay, so tengok nombor 3 pula. Once it reach to the end of endoplasmic reticulum, the membrane buds off to form transport vesicle. So maksudnya adalah kita boleh cakap rough endoplasmic reticulum tu dia akan hasilkan transport vesicle. Why? Because they want to deliver the protein to the Golgi apparatus. Tengok. The transport vesicle contain protein, move towards Golgi apparatus and fuse with it. Maksudnya transport vesicle ni dia akan bergabung dengan Golgi apparatus. Supaya apa? Supaya protein tu boleh masuk ke dalam Golgi apparatus. So awak tengok. Protein tu warna kuning kan. So dalam ni dia akan meresap masuk ke dalam ni lah. So apa Golgi apparatus tu buat? In Golgi apparatus, the protein are modified into enzyme. So maksudnya bila sampai dekat sini, Golgi apparatus tu akan menukarkan. Modified tu maksudnya ditukar lah. Ataupun dibuat lah. Modified, ubah suai protein tu kepada enzim. Lepas tu sekarang ni kita dah hasilkan enzim, betul tak? So tengok Golgi apparatus ni dia akan hasilkan secretory vesicle. Macam tadi rough endoplasmic reticulum dihasilkan transport vesicle. Tapi kalau Golgi apparatus ni dihasilkan secretory vesicle. Apa fungsi secretory vesicle tu? Adalah to deliver the enzyme. Sekarang kan Golgi apparatus kan dihasilkan enzim. So enzim tu kita nak hantar dia keluar, betul tak? Sebab dia extracellular kan enzim yang digunakan di luar sel. Okay, so the secretory vesicle will carry the enzyme to where? To the plasma membrane. Sebab apa? Plasma membrane kan adalah lapisan yang paling luar kan dekat sel tu. So tengok, the secretory vesicle that bring enzyme will move towards plasma membrane and fuse with it to secrete extracellular enzyme. So kalau awak tengok kat sini, secretory vesicle yang bawa enzim tu dia akan fuse. Fuse tu maksudnya dia akan bergabung dengan siapa? Dengan plasma membrane. So the enzyme can be expelled out. Sebab apa kena expel out? Sebab enzim tu digunakan di luar sel. Okay guys, so this is how it works. Okay. Okay, seterusnya adalah you should know about mechanism of enzyme reaction. Okay, remember the mechanism of enzyme reaction is influenced by lock and key hypothesis. Okay, lock and key hypothesis. Apa maksud dia? Okay, so maksudnya adalah reaction enzyme ni adalah berdasarkan lock and key hypothesis yang dicetuskan oleh saintis. So kita tengok mana ni? Ni, the binding of molecule substrate on active site of the enzyme is a specific like a lock and key combination. The enzyme is represent as a lock. So enzyme ni sebagai lock. Okay, substrate ni sebagai key. Tengok. Okay. Uh, okay, so the enzyme. Okay, enzyme tu adalah log, substrate tu adalah key. Okay, sekarang ni cuba awak tengok ada dua substrate kan. Yang mana satu yang berjaya combine dengan enzyme. So mestilah yang ini kan, yang ini dia saja nak tunjuk kepada awak. This want to show to you that the reaction of enzyme is specific. So maksudnya hanya substrate yang tertentu saja boleh combine dengan enzyme. Yang ni tak boleh lah sebab apa? Bentuk dia tak sama. Dia kan active site dia kan. Remember, enzyme act as a lock while substrate as a key. Okay, so pertama sekali substrate tu dia akan bind to the active site of the enzyme. Lepas tu this will produce enzyme substrate complex. So what happen? The enzyme will break the bond in the substrate. Therefore, the products are produced and the enzyme can be used for the next reaction. Boleh faham? So, awak tengok kat sini ya. Okay, boleh faham so far? Okay, yang ni, awak tengok. This is the graph to show to you if you use enzyme, if you don't use enzyme. So, kalau yang warna merah ni menunjukkan if you use enzyme. Okay, warna biru ni menunjukkan jika awak tidak menggunakan enzyme. So kita tengok this is reactant. Reactant tu maksudnya adalah apa yang awak gunalah. Sebagai contoh awak guna enzyme dan juga substrate. So those thing we known as a reactant. Produk ni yang kita hasilkan tu. So kalau awak tengok kat sini, kita mesti nak menghasilkan produk dengan cepat, betul tak? So, kalau kita tengok yang merah ni yang menghasilkan produk dengan lebih cepat. Sebab apa? Awak tengok lengkungan dia sekejap saja. Terus jump into produk. Tapi yang ni kena naik atas dulu, lepas tu baru turun. So, it takes longer time. So, that's why activation energy with enzyme is much faster. Okay? Most of reaction in the cell require high activation energy. Activation energy is the energy needed to break the bond in a substrate molecule before reaction can occur. The enzyme function by lowering the activation energy. So bila awak guna enzyme, you can lower the activation energy. So maksudnya tak perlukan banyak tenaga lah. Kan kita kan nak save tenaga untuk buat benda lain kan, betul tak? Okay. So if you don't use enzyme, your activation energy is higher. So you just waste your energy there. Okay, so when you use enzyme, the enzyme will lower down the activation energy. So less energy is required to break down the substrate into product. Okay. Okay guys, so next we are going to study the factor that affect the enzyme concentration. Okay, ada berapa ni? 1, 2, 3, 4. So there are 4 factor that can affect the reaction of enzyme. Okay, are you ready? Okay, let's see what else. Tak ada dah. Okay guys, so let's continue. Okay, now we are going to focus on 4 factors that can affect the enzyme reaction. Okay, the first factor is the effect of temperature. Okay guys, so remember for the factor, it is always at the x-axis. Axis X ni, paksi X ni. Okay, and paksi Y dia ni adalah rate of reaction of enzyme. Okay, so this is temperature. Okay, so let's go one by one. Okay, number 1, okay, at low temperature. Okay, so here you know this is 0 temperature, right? So this is 10, 20 until 60. Okay, at low temperature, the rate of reaction catalyzed by enzyme is low. Okay, so When the temperature rise, the kinetic of the substrate molecule and enzyme also increase. And this increase the frequency of effective collision between substrate molecule and enzyme molecule. Thus, the rate of reaction between enzyme and substrate will increase. With every rise of temperature of 10 degrees Celsius, the rate of reaction controlled by enzyme will double until it reach optimum temperature. Okay, so I want to conclude here is that the higher the temperature, the higher the rate of enzyme reaction. Why? When we increase the temperature, we will give kinetic energy. We will give kinetic energy for enzyme and also substrate to collide. So bila dia berlanggar kan, bila berlanggar enzyme dengan juga substrate, this can increase the possibility for the substrate to bind with the enzyme in the active site. So this will increase the rate of reaction until it reach optimum temperature. So number 2 ni, if you notice, the rate of reaction is the highest. Betul tak? Paling tinggi, paling maksimum. Betul tak? Ni kat kemuncak. So we can say that here 30 degrees Celsius is optimum temperature. Optimum temperature tu apa cikgu? Optimum temperature is the best temperature that can contribute to the highest rate of enzyme reaction. Sebab cara tengok nombor 2 At optimum temperature, the enzyme reaction is at its maximum. Okay, the optimum temperature for enzyme reaction in human body is 37 degree Celsius. So if you want to have the highest rate of enzyme reaction, you should use optimum temperature, which is 30 degrees Celsius. Okay, so what happen if we keep increasing the temperature beyond optimum temperature? Okay, apa yang akan jadi kalau kita sini dah elok dah, tiba-tiba kita naikkan lagi temperature, apa akan jadi? After reaching optimum temperature, a further increase in temperature will reduce the enzyme activity rapidly until it stop at 60 degrees Celsius. So kalau awak tengok lepas 3 degree Celsius melebihi 37 degree Celsius, the rate of reaction start decreasing and finally stop at 60 degree Celsius. Sebab apa ni? At this temperature, the enzyme become denatured. The enzyme become denatured as the chemical bond in enzyme molecule break at extreme temperature. Cikgu, apa maksud denatured? Okay, denatured is condition. Denatured is a condition where the active site of the enzyme is damaged. Okay, cikgu ulang Denatured is a condition where active site of the enzyme is damaged. So bila active site damaged, can the enzyme bind with the substrate? No. Sebab itulah rate of reaction start decreasing. Okay, the enzyme are unable to retain the 3D dimensional form. The active site of the enzyme change. Therefore, the substrate cannot complement to the active site of the enzyme. So, kalau awak tengok dekat 60 degree Celsius ni, all the enzyme are denatured. All the enzyme are denatured. Sebab itulah rate of reaction 0. Betul tak? So, kalau kita guna 60 degree Celsius, rate reaction 0. Sebab apa? All the enzyme are denatured. Sebab dia punya active site semua dah rosak. Sebab apa? Sebab bila kita guna higher temperature, active site tu akan damage. That one is in term of temperature. Now, we go for the effect in terms of pH. Okay guys, so same here. Awak tengok kalau part CX ni, they use pH. Okay, part CY, rate of reaction. Okay, so I want to emphasize here, every enzyme has its own optimum pH. Macam tadi optimum temperature, betul tak? Optimum temperature are the same, 37. Tapi untuk pH it's different according to what enzyme you use. For example, kalau pepsin ataupun protease. Ingat pepsin dengan protease benda yang sama, fungsi yang sama. Okay, protease ni dia punya optimum temperature is pH 2 which is acid pH. Okay, tapi kalau amylase, tengok ni, amylase and most enzyme. Okay, pH dia adalah 7, pH neutral. Tapi kalau trypsin, apa lagi, lipase, sebagai contoh, dia punya pH adalah pH 8 which is alkaline. So every enzyme has different pH, optimum pH. Kalau protease ataupun pepsin, they are good in acidic medium. Kalau amylase ataupun most of the enzyme, they are suitable under neutral condition. Tapi kalau trypsin dan juga lipase, they are good under alkaline condition. So what happen? Okay, kita pergi satu-satu. Okay, the enzyme activity is influenced by pH of surrounding solution. Generally, all enzymes react most effectively at their optimum pH. So kalau pepsin, pH 2 lah. Kalau amylase, pH 7. Kalau trypsin, pH 8. Most of enzyme are most active in range between 6 and 7. For example, salivary amylase works at the pH 6.7, 6.9, 6.8. So 7 lah tu. However, there are some exception. For example, pepsin enzyme in the stomach, okay, at optimum pH range between 7.5 until 7.2.5. So maksudnya acidic lah. Trypsin enzyme in duodenum, on the other hand, only works well in alkaline medium, with a pH around 8.5. Okay. Okay guys, so if you notice, okay, ni cikgu nak, okay, for example lah, cikgu ambil satu contoh Okay, kita ambil pepsin ni. Okay, so macam mana explanation dia? The higher the pH, the higher the rate of reaction. Betul tak? Ni awak tengok ni The higher the pH, the higher the reaction. But at optimum temperature, which is 2, at optimum temperature, which is 2, the rate of reaction is the highest ataupun maximum. Betul tak? Tapi beyond the optimum temperature, the rate of reaction start decreasing. Betul tak? Okay, so kenapa dia beyond optimum temperature, it start decreasing? So apa ni explanation dia? The change in pH value change the charge of H plus. H plus tu hydrogen ion lah. Okay, the change in pH change the charge of ion H plus of the active site of the enzyme and substrate surface. As the result, enzyme substrate complex cannot be formed. When the pH of environment returns to the optimum level, the charge of positive site will be restored. The enzyme will return to function normal. The extreme change in the pH value can break and change the active site of the enzyme. So remember, you have to emphasize the word active site. So when there is a change in pH, this will affect the structure of active site. So bila active site tu berubah, this can prevent the substrate from binding with the enzyme. The next factor is substrate concentration. Awak tengok graf dia lain kan? Macam tadi, graf untuk temperature macam ni. Graf untuk pH macam ni. Graf untuk substrate macam ni. Okay, awak tengok Okay, sama juga part C S dia substrate concentration. Part C Y dia adalah rate of reaction. So apa jadi awak tengok nombor 1. Okay, if the concentration of enzyme is fixed, when the concentration of substrate increase, the rate of reaction controlled by enzyme will also increase, leading to an increase of product created. So ini adalah substrate, betul tak? Substrate concentration. Okay, tapi concentration enzyme kita sama, kita fixkan. Tapi kita tambah concentration of substrate. So awak tengok okay, nombor 1 ni expansion dia. When substrate concentration increase, the opportunity of effective collision between substrate and enzyme molecule also increase. Betul lah. Okay, so bila substrate concentration increase, so banyaklah collision between substrate and enzyme. So bila collision enzyme dan substrate berlaku, rate of reaction akan increase. The reaction rate continue to increase until it reach maximum level. Ni maximum level dia. The rate of reaction is constant. Sebab apa? Ni kita tengok ni. Nombor 2. Kenapa rate of reaction ni dia constant? At maximum level, the concentration of enzyme is the limiting factor. Sebab apa? Kita increasekan substrate, tapi concentration of enzyme kita sama. So maksudnya kita punya limiting factor kita adalah concentration of enzyme, because enzyme is the one that has active site to change, okay, the substrate into product. The rate of reaction can increase when the concentration of enzyme increase. So kalau awak nak naikkan rate of reaction, you need to increase the concentration of enzyme. After reaching maximum level, all active site of the enzyme are saturated. Maksudnya at maximum level ni, maksudnya all the enzyme are fully bind, okay, with the substrate. So no enzyme are available, okay, and are involved in catalytic reaction. Maksudnya kat sini adalah kita punya enzyme lah limiting factor, sebab semua enzyme tu sedang bekerja, sedang combine with the substrate. So substrate yang lain tu tak dapat combine lah. Okay, that's why the rate of reaction become constant. Okay, that's all. Okay, and then the last one, the effect of enzyme concentration. Tadi substrate concentration. Yang ni kita nak cerita pasal enzyme concentration. So, awak tengok, this is the graph. Macam tadi, graph dia macam ni. Tapi kalau enzyme concentration, graph dia macam ni. Okay, awak tengok nombor 1 ni. When the concentration, okay, part C X dia enzyme concentration, part C Y dia adalah rate of reaction. Okay, nombor 1, when the concentration of enzyme increase, the rate of enzyme reaction will increase. Betul lah. Because the presence of more active site are ready for catalytic reaction. So, when you have more enzyme, it means that you have more active site available to bind with the substrate. Sebab itulah rate of reaction naik. But, okay, nombor 2 ni awak tengok ni. Ni apa jadi kalau kita teruskan increase? Apa jadi? Okay, if the concentration of enzyme is double, the amount of substrate converted to products per unit of time is double with the condition when there is excess supply of substrate. So kalau awak nak keep increasing the rate of reaction, kalau yang 3 ni dia constant, kalau yang 2 ni awak nak terus naik, betul tak? So if you want to have this kind of condition, you should add more substrate. Awak kena naikkan substrate lah. Okay. Tapi kalau awak tak naikkan substrate, this will happen. The maximum rate, at maximum rate, the concentration of substrate is limiting factor. Betul lah. Macam tadi, kalau yang ni, dia punya enzyme limiting factor. Kalau yang ni awak tukar je. Substrate is the limiting factor. Because all the substrate are combined with the enzyme. So, that's why our substrate is the limiting factor. The rate of reaction can only be increased by adding more substrate. Okay. Okay, guys. So this one, this one we are going to do an experiment. Okay, when you when you come back to the school, okay, we are going to do this experiment. Okay. Okay guys, so this is the last subtopic for today. So it's about application of enzyme. So this one, I think you can read on your own. Okay, it's very easy. So here, okay, I will paste the information in the Telegram group so you can read on your own, because this is a very straightforward information. Okay, guys, so I think that's all for today. So as a homework, okay, please try to complete, okay, the enzyme subtopic on bioscore. Okay, so on the next class, we are going to discuss, okay, the exercise that you have done in bioscore. Okay, so I see you next time. Assalamualaikum warahmatullahi taala wabarakatuh. Bye bye.